Let's quickly go through DNA, proteins, amino acids, specimen 0.5. Let's have a little look at this. So we've got 3 different amino acids joined together. We've got peptide bonds, C double bond O NH, formed when a carboxylic acid ending COOH reacts with an NH and forms water in a condensation reaction, and leaves CONH as a peptide bond between those 3 amino acids. If you break it up using concentrated hydrochloric acid will hydrolyze, put the water back in and break the bond, and break them back up into the amino acids. You've got to be able to work out once breaking those amino acids, breaking the peptide bonds, that the CO turns back into a COOH and the NH turns into an NH2. If it asks for the Zwitter ion has got 1 NH3 plus and 1 COO minus, just one of each, because at a certain pH called the isoelectric point, the COOH loses an H plus, and that joins onto the NH2 to make NH3 plus, a Zwitter ion. If you're asked for something then at the low pH, which is acid, acids have got H pluses. Acids make H pluses, donate H pluses. So all of the NH2s become NH3 pluses, all of them at a low pH. At a high pH, the opposite happens. The COOH loses an H plus because at high pH you've got a base. Bases react with acids, so it reacts with the COOH and removes an H plus and makes COO minuses, and all of the COOHs become COO minuses. Let's have a look at set 2. So let's have a look at 2017. Remember your diagrams. Remember that you've got in a nucleotide you've got a base and 2 deoxyribose and a phosphate group. It's got 3 parts: phosphate, 2 deoxyribose and a base. The base joins onto the 2 deoxyribose at the point where it says X, and your bases then join up 2 nucleotides with a hydrogen bond between the bases. You've got to make sure you know which base reacts with which base, and make sure you know how they hydrogen bond. So if you wanted to, on your data book, put the lone pairs of electrons on the O's, the lone pairs of electrons on the N's, make the NH and the NH's NH plus, and then join up with hydrogen bonds on your data sheet, ready to be able to answer the question between the lone pairs of electrons on O's and the H pluses on the NH delta plus, hydrogen bonding. Your complementary bases, so make sure you know which your complementary bases are. Guanine and cytosine will form 3 hydrogen bonds, and adenine and thymine will form 2 hydrogen bonds. When you're asked to join them up, you have to flip, so reverse, mirror image left to right the base, so that the hydrogen bonds align. Otherwise they're going to go round the other side of the base and not be able to form straight lines. So they should be straight lines like on the diagram, but to do that you have to flip your base over. 2018. So here's one where you just have to draw hydrogen bonds and say that it's between the delta plus on the H, which attracts a lone pair of electrons on the O, straightforward. And then you've got cysteine. Cysteine forms sulfur to sulfur bridges, where the SH part on one cysteine can bond with the SH part on another cysteine, and it forms an S to S bond, the sulfur-sulfur bridge between the 2 bases. 2019, it might ask you to name the 3 constituent parts of a nucleotide, in which case you'll have to ring the phosphate, the 2 deoxyribose and the base joined to it.